Okay, it's with great pleasure to announce Eva Dyer, who will give the next talk. It will be about quantifying mesoscale neuroanatomy with X-ray microtomography. And I should first give her great thanks for speaking, because she was graciously willing to take over that slot for us to cancel after this.
Thank you to the organizers for inviting me. Oh wait. Okay, yeah, so I'm going to be talking today about some work on quantifying mesoscale neuroanatomy, and the particular tools that we're using are X-ray micro-CT. So to motivate this, much of what we know about neurocomputation is based upon studies from small numbers of neurons. However, there's 70 million neurons in the mouse brain and over 100 billion neurons in the human brain. So the hope is that with large-scale data sets that provide us access to larger numbers of neurons or larger brain volumes, we can start to answer a number of new questions that we can't by studying just small numbers of neurons. So in particular, we're interested in questions like, how are neurons wired together, especially in large-scale circuits? How might the wiring and computation vary across different brain areas? And that's something that you need to be able to access large volumes of brain to really understand and assess. And also what happens to these circuits and to these different brain volumes in the face of disease, aging, or other learning. So in order to kind of face this issue of needing larger brain volumes, there's been a recent neural data deluge. So I'm going to quickly go through a few different types of modalities that have provided us with unprecedented amounts of information, both at very small scales, so at nanoscale lengths, and also at mesoscale, which is going to be the focus of my talk later. So just to set the stage, electron microscopy is a technique that's now become pretty prominent in neuroscience. Electron microscopy provides us with very high resolution data, so the resolution is about 10 nanometers per voxel. The volumes that people can currently image with electron microscopy is something on the order of a cubic millimeter, so a very tiny fraction of the brain. But within that cubic millimeter of data, which takes approximately 3 months to image, you get about 10 petabytes of image data. So it's this very large-scale problem in terms of the amount of data that you can collect with electron microscopy. And then some of the most sophisticated reconstructions that have been able to been carried out. So doing full saturated reconstructions of cubic millimeters or even smaller, something like here where we actually look at all of the processes, all of these different colors represent different neurons that they've been able to trace out within these electron microscopic data sets. The diameter of this volume is about 7 microns, and so within this amount of data, to segment it, it took about 6 years to do a full saturated reconstruction. There are methods that are able to get sparser subsets of these types of volumes on the order of months or even years, but it's still a very challenging problem even for sophisticated nonlinear techniques like deep learning architectures for connectome reconstructions. And so what you can see in electron microscopy data is basically everything up to the nanoscale. So you can see axons, so the outputs of neurons trying to connect to other neurons, the dendrites of neurons, so their input terminals, and you can also see individual synapses, which give you a measure of the connectome or the connections between these different neurons. So at this scale, we can see a lot of stuff. In addition, there's been a number of new techniques which use serial 2-photon tomography, which is a technique to basically reconstruct, at this scale, entire whole brain circuits. And so the typical resolution for serial 2-photon tomography, I gave away my... results. I got to the answer to the question. Okay, so with serial 2-photon tomography, you can see things on the order of a half a micron. You can image an entire mouse brain, which is 400 cubic millimeters, in about a week. And what this generates is on the average of 30 terabytes of data. And so what you can see from these, so what I'm showing here is a slice from the Allen Institute Mouse Connectivity Atlas. And what we're showing here are basically injections of a virus that are placed into different brain areas. And what you can see are the projections of all the neurons that are targeted by that virus and where they project to. And so if you use clearing approaches to basically melt away the rest of the brain, what you can get out with these types of serial two-photon tomography techniques are entire reconstructions of whole brain circuits. So this top one here is from the Allen Institute using similar techniques, and down here this is a figure from a paper from Janelia. And so with both of these techniques, you're able to see the extent of axons and circuits over whole brain volumes. And what's visible are on the order of tens of neurons, and you can see both the local and the global connections with these types of methods. And so what I'm going to talk about today is a slightly different approach that we've been using, and it's complementary to these other types of modalities that I just described. And so in particular, what we're using is X-ray microtomography, which I'll get into the details of. What we're able to see with this are all of the cell bodies, so both neurons, also glia, interneurons as well. We're able to resolve the blood vessels as well, so it gives us information about both the vasculature and the cell bodies within the volume. And we're also able to resolve large axons and dendrites. And so in order to collect this data, to get an entire shrew brain, which is 80 cubic millimeters at 0.65 microns resolution, takes about 6 hours. And the cool thing about X-ray is that you don't have to actually section the tissue. So X-rays can penetrate through much deeper volumes, and so it allows you to get a full 3D reconstruction of a brain volume without having to cut the tissue and stitch it back together in the end. And so the types of things that we can reconstruct here are these dense reconstructions of mesoscale neuroanatomy. So you guys have already seen this, but this is an example of the type of reconstructions that we're able to get after running these data sets through the pipeline that I've developed and will describe. So here what I'm showing is about a cubic millimeter of brain tissue from mouse somatosensory cortex. And so in red here, I'm showing all the blood vessels that we've reconstructed with our technique. And then on top, there's a small little volume of cell bodies which have been manually segmented. And so I wanted to show this to start off just to get a sense of the scale of this type of problem. So with just a cubic millimeter of tissue, we get about 100 gigabytes of data. And from that, in order to manually segment this 200 micron cube of cell bodies, takes about 2 weeks of person hours. So this is an extremely time-intensive process, and one that really requires automated techniques in order to parse through these large volumes of data. And so here's some more reconstructions from our pipeline, and here I'm showing you exactly the same volume, where you see this small little tiny cube of segmented data, and then all the rest of this that's filled in is about 50,000 cells that we've detected with our algorithms, and here I'm just visualizing all the individual cell bodies that are detected. So now I'm going to kind of give some insight into the method and talk about the ways in which we're able to develop these pipelines to analyze these large data sets. So the first step is to acquire the data. So this is just a schematic showing the method. So basically it works similar to normal X-ray techniques, where you want to shoot X-rays at a sample and then measure multiple angles or projection measurements. And so you do that by first cutting out a small cube of brain tissue. We actually embed it in plastic, and then basically the X-ray source hits this sample of tissue, and we get a certain measurement that comes out. So what happens is the X-rays can be converted into visible light through the use of a scintillator crystal. And so each of the measurements that we collect is a single angle of this sample, and we measure how many X-rays are absorbed by the sample and how many are incident on the detector. And so then we're going to rotate this sample around, and effectively we can collect a cubic millimeter of data in 6 minutes in comparison to 3 months with electron microscopy. So it's a very fast and high throughput method. So what comes out are images, a 3D stack of images, and this is one example of a microtome. So what I'm showing here is a slice of brain tissue where we can see individual cell bodies as these light gray, and we can also see blood vessels as these dark spots in the image. And so if we zoom in closer, we can see a number of different things. We can see, the cell bodies along, and I showed before, these blood vessels. In addition, we can see apical dendrites, so we can see some large processes that are coming from certain neurons in the volume, which give us some insight into both the identity of whether or not they're a neuron or an interneuron, as well as some information about the type of neuron that it could be. So what's really cool about this method is because we can do X-ray without sectioning the volume, as is common with optical methods, we can do correlative imaging with EM right after the X-ray. So what I'm showing here is an example where we have an X-ray volume and we've identified a subset of neurons that we can then do subsequent electron microscopy analysis on, and then we can actually identify and register the two volumes onto one another. So what this provides us with is it gives us a way to do non-invasive imaging at the micro scale, and then go in later and then study its connectome or study nanoscale properties of the same sample. So we have two modalities overlaid on the same brain result. So in order to, as I mentioned before, we want to be able to develop automated algorithms that can parse through these large data sets at scale. And so the first thing that you would do if you're going to segment this data in order to find the blood vessels and the cell bodies is to simply threshold the data. That's the simplest path. So what happens is when you threshold the image data, it comes up like this. The issue is that with X-ray tomography, there's a lot of local gradients that are present in the images that basically, if you use the intensity alone, you lose a lot of the structure in the sample. So we had to develop more sophisticated methods, which, if we didn't, I wouldn't be up here today. So. Okay, so in order to do that, I developed a pipeline for processing these large volumes. We have this open and available on GitHub. It consists of three main components. First is segmentation, so being able to separate the cells and the blood vessels from the background. In addition, we also need to be able to find the position and the diameter or the radius of cells, and so for that we've developed an algorithm to actually perform cell detection, which is separate from the segmentation step. And then finally, we developed a number of tools to basically optimize our workflow in order to find the right set of hyperparameters to both validate and improve the performance of the algorithms. So the first step is to segment, and so as I said before, we want to separate the cells and the vessels from the background. So to do that, we applied a random forest classifier. And so what I'm showing here is just the original CT slice, and then basically after applying a random forest classifier on a number of kind of standard computer vision features applied to these images in 3D, we get out two different probability maps. First is the probability that any voxel is a blood vessel, and then we also have the probability that any voxel in this volume is a cell. And so these are just these two different probability maps that we extract from these data sets after running our classifier on it. And so if we kind of zoom in a little more, we can see a bit more detail. So this is the original image slice, and these are our ground truth labels that we've been able to collect. And so here I'm just showing the output of our cell finding algorithm and our vessel segmentation algorithm. And we see both qualitatively and also through validation that this actually does quite a good job of identifying the position and size of cells and the vasculature in the sample. The issue, however, is that when you take if you were to just threshold this probability map on the cells, cell bodies are so closely connected to each other, or they're so close in space, that oftentimes you get these large clustered components of cell bodies, and so we need to be able to segment those out and separate them to find individual cells. So to do that, we developed an algorithm, which is a greedy approach, which was necessary in order to really apply this at scale. And so the basic idea is to apply a spherical template to the volume, to sweep it over this entire connected component, or in our case, this probability map, find a cell that produces maximum correlation, and then basically remove that, and we keep on iterating until we've selected all the cells within the volume, and we reach a particular stopping criteria. So this is an example of where, after we've iterated, we finally finish by finding the last cell, and then we reject this final vessel component because it doesn't look like a sphere or fit our spherical shape. Okay, so after we've gone through the steps of segmentation and also cell detection, the next step is to be able to optimize our methods. And so in order to do that, we both collected a number of dense training data sets that were used to validate our methods and also to tune hyperparameters. And then we just have some brute force search, and as well as some more sophisticated search algorithms to be able to tune the hyperparameters in order to maximize precision and recall of these segmented volumes. And so these are some examples of the manually annotated data sets that we've collected. As I mentioned before, actually collecting these data sets is very challenging. And so this is a very labor-intensive process, which is one that we want to try to avoid in the future. And so first we collect these training data sets, and then what we do is, for both the vasculature and the cells, we basically sweep over a number of different hyperparameters, and then find the set of hyperparameters that basically maximize these different precision and recall metrics. And so, based upon those parameters on a training volume, on this large training volume here, we then apply that algorithm at scale over the entire volume, and then we can evaluate the reconstruction performance on these held out test data sets. And so in the end, on these three different, so this is the training cube, and then we have two test cubes, we get on average about 85% accuracy, although in some cases we get slightly better precision. And so you can see there's, while this is the first pass at doing something like this on X-ray microtomography data, we see that we get actually pretty accurate results, but there's still some gap in terms of improvement that we'd like to address with more sophisticated classification algorithms. So these are some results from the final cubic millimeter. Here I'm showing all the detected cell bodies, we have all the vessels, and then the two combined with each other. And you see really just the scale and the number of cells that are present within just a tiny portion of the cortex. So in addition to providing tools for segmentation and cell detection, we also developed a number of tools for computing spatial statistics over these large volumes. So what this involves is, mainly we want to get estimates of the cell density over the volume, and we want to do it in such a way that we can actually measure the 3D distribution of cells in an unbiased way. So in order to do that, we apply a nearest neighbor based approach for density estimation. And so the idea is that the density at each point of space represented by this vector v is inversely proportional to the distance that from each point to its kth nearest neighbor. So if there's a very high density of cells, then the distance to the kth nearest neighbor is small, and so this density estimate will be large. And so we apply this, and it's nice because it's scalable, so you can just basically compute nearest neighbors over this volume. And so out of that, we're able to get estimates of both cell size using our cell detection algorithm, the cell density as well. And what's nice is that the range of these estimates do align with previously reported results, which gives us further confirmation that this method works even at large scales. And so the mean is like on average with what people report in mouse cortex. In addition, we can also measure the cell to vessel distances, and so in this case we can understand things about how how the vasculature is interconnected with higher or lower density areas of cells. Okay, and so to accompany these tools for spatial statistics, we also developed a number of tools to visualize and overlay these different visualizations on the volumes. And so here I'm just showing some visualizations in 3D. What we're able to see is that we can really nicely preserve the three-dimensional structure of the tissue in a way that's not often possible when you have to section and then stitch things back together. So X-ray can be very valuable in situations where you want to preserve this dense 3D structure of the anatomy. And so we have tools then to also overlay the density, the cell density estimates that we got from the last step onto the appropriate volumes, and we have visualization tools online to actually look at the reconstructions and results. Okay, so what I've described is, being able to apply relatively simple classification tools, but to very large volumes of data. So, you might argue that 100 gigs isn't a large-scale problem, but as we start to scale this up even further, namely to entire mouse brains, and we're also working on Etruscan shrew brains, which are much smaller. So in order to scale this up to an entire mouse brain, it will require parsing about 20 terabytes of data. And so the way that we had to go about solving this problem was to make sure that our workflow was scalable and could be applied to larger data sets as they come online. So the tool that we currently use but are extending is to use a tool called Lonnie, the Lonnie pipeline. It provides a way of creating a distributed workflow, and then basically Lonnie will distribute your jobs automatically. And so what you're able to do with this tool is you create your workflow in this kind of graphical environment. So these are all the different steps of our pipeline. Within each of these, we just have a call to a Python or a MATLAB function. And then basically, if you feed in a certain amount of data to this, it figures out, once certain jobs are done, how to distribute the rest. And so this provides a tool to basically start to scale these things up. And so running about a cubic millimeter, so 100 gigs of data, takes about 8 hours on an 8-core machine, so about 64 core hours. And so we're currently working on ways to actually place this on supercomputing resources at Argonne National Laboratory, where we're collecting the X-ray microtomography data. And so this is just an example of some recent results. This is a panorama where we've stitched together multiple different views of an entire mouse brain. In this case, the data set that was collected was about 6 terabytes. And here I'm just showing the projection data. And so what you're seeing are the actual X-ray projections that are collected on this detector that I showed. So this is before actually doing the tomographic reconstruction. And so this is just a 3D view of this stitched volume. And so what you're able to see here is just the gross anatomy of an entire mouse brain. Recently, however, we were able to actually apply this technique. Let's see if this works. Okay. I just want to show you this video. Okay. So what I'm showing here is a pass. So I'm going through an entire mouse brain volume. This is, oh, I'm not. Okay, so this is a sagittal view of the mouse brain, and so what we're seeing here is a reconstruction of the entire brain. So right here we start to see a lot of myelinated axons that are coming through. This is the cortex. All these white, bright streaks coming through are all blood vessels, and so we're able to get This is currently at 4 microns resolution, but we're able to image an entire brain without sectioning it at a micron resolution. And then here I'm just downsampling as well. So this is a pretty exciting advance because we're finally able to image an entire brain in a dense way without having to section the tissue. Okay. Okay, so some of the future work is being able to apply deep networks or deep learning to be able to solve this segmentation problem. As I showed, there's a bit of a gap in the amount of segmentation performance that we can improve upon. And so, yes, we're currently working on that. In addition, using things like transfer learning to take this really expensive manually annotated data, train a classifier, and then be able to transfer that learned model onto a new data set that we haven't seen before. So in summary, I've described a new pipeline that we've developed for large-scale neuroanatomy from X-ray, micro-CT. I've described some of the algorithms for segmentation and cell detection that we've developed, as well as tools for computing and visualizing spatial density estimates. And of course, this work couldn't be done without a large team of people. So Argonne, Bobby Kasturi, and a number of other beamline scientists, and Chris Jacobson from Northwestern have all been instrumental in being able to collect this data and really scale this up to the types of volumes that we're looking at. Our team at Hopkins, through NeuroData, have been contributed to a lot of the distributed workflows, as well as visualizations. And then this is also work with my postdoc supervisor, Conrad. And so with that, I'll open it up for questions. Thanks.
So do you also have plans for detecting dendrites and synapses?
Yes, so at this particular resolution, we can't see individual synapses. There's ways to still use CT, but at the nanoscale. And so there's some efforts going the nano-CT route. So there's two different approaches. One is scaling up at the micron scale, and the other is doing nano. And so currently with the data set that I just showed on the whole brain, we can see what seems to be individual axons. And so a big push within the lab is being able to trace out myelinated axons over very large regions of the brain.
So if you had a transgenic that could put in some dense label synapses, a little punctum, you might be able to do that with comfort.
Yeah, I mean that could be approach. Currently, X-ray histology is still relatively underdeveloped in comparison to optical techniques. So a lot of the transgenic tools that we have in optics aren't currently applied, but there are ways to use apex or other types of stains to turn certain types of neurons, as having a more intense contrast. And so we are looking at those types of histological approaches for X-ray to help with that. Yeah.
Fascinating work. Thank you so much. I was wondering, you have two imaging modalities, two kinds of data that you're getting out here, the cell bodies, stomata, and the vasculature. So have you noticed any kind of correlation, for instance, between the density of vasculature in a particular region, density of cell bodies? I could imagine that working both ways, maybe there's more vasculature, so maybe there's less room for cell bodies, but also one of the primary assumptions behind fMRI is that more vasculature has various connections to, or at least more vascular activity has some implications for neural activity. So have you noticed anything like that?
So with the current techniques where we're not able to see the difference between, say, glia neurons or pyramidal neurons and interneurons, we haven't seen any really surprising things. So the vasculature seems to be relatively homogeneous relative to the cell densities. But so there are, potentially when we have ways of staining different types of neurons, those differences might pop out more. But yeah, so we're currently doing some studies in barrel field and somatosensory cortex, where they find that cell density should be roughly correlated with higher blood vessel density outside of the barrels, and so we're looking at those kinds of systems as a way of potentially testing those questions. So that's a good question.